Okay, in honors biology, we're going to be covering chapters 10 and 11, evolution of populations together. This is a topic that you might want to make sure you go back and read the chapter 10, 11 in your book. It's not my forte, and so you'll get a better picture by reading the book. So evolution is sometimes said to be descent with modification. And there are several terms that go along with evolution. Phylogeny, Phylogeny, is the evolutionary history of an individual. Catastrophism is a principle that events in the past occurred suddenly, so some catastrophe happened and were caused by mechanisms different than those operating presently. So that was one of the theories of evolution from a long time ago. Uniformitarianism was proposed by Lyell, and it's a mechanism of change that are constant over time. So there wasn't a sudden event, it's just been a constant mechanism that's affected populations. He incorporated James Hutton's philosophy that Earth's geological features could be explained by gradual mechanisms still operating today, and that is called gradualism. So you have catastrophism, you have uniformitarianism, and you have gradualism. Now, the theory of natural selection is a process, and you're going to have to write quite a bit here. It's a process in which individuals that have certain heritable traits survive and reproduce at a higher rate than other individuals because of those traits. So they have inherited certain traits that makes them better to adapt. Over time, natural selection can increase the match between an organism and its environment. If an environment changes or if the individuals move into a new environment, natural selection may result in adaptation to these new conditions, sometimes giving rise to new species. Individuals do not evolve, but a population can over time. So evolution doesn't affect individuals, it affects populations. It does not act directly on individual genes, but it enables individuals who express favorable traits to reproduce and pass those traits on to the offspring. So it works on phenotypes, not genotypes. So it works on physical characteristics, not on genetic makeup. Natural selection can amplify or diminish only those heritable traits, so those that are inherited, that differ among the individuals in the population. Organisms share characteristics because if it's because of common descent, we call that homology, or because homo means the same. For example, mammal forelimbs. Or because natural selection affects independently evolving species in similar environments in similar ways, and we call that convergent evolution. So convergent evolution is the independent evolution of similar features in different lineages, which leads to analogous structures. And analogous structures are those that share a similar function, but not a common ancestor. So like wings of a bird and wings of an insect. They don't have common ancestors, but they have a similar job to help an animal fly. So here's our example. A bat's wing is analogous but not homologous to a bird's wing. Whereas homologous features share common ancestry but not necessarily similar function. So this is because of a common ancestor but don't necessarily serve the same function. Okay, vestigial structure. So like for instance, your appendix is a vestigial structure. They no longer have a function in that organism. So places that you have to write a lot, obviously you might want to put it on pause and stay caught up. Okay, population. So let's define population. So you're going to fill this all in. All organisms of one species that live together in one place at one time. So population are all the organisms of one species that live together in one place at one time. The gene pool are all copies of every type of allele at every locus in all members of that population. So that's the gene pool available to the organisms. This leads to genetic variation. Allele frequency is how often an allele appears in a population. Biological fitness refers to the organism's reproductive success. It is measured by its contribution to the gene pool of the next generation. Okay, so genetic variation in a population comes from several things. Number one, it can come from mutations. That's just a random change in the DNA that did not get caught. So one base was substituted for another. Recombination occurs during meiosis, so it's a shuffling of genetic material, like in prophase 1. Okay, natural selection can affect a population and the distribution of a trait. So microevolution is evolution on its smallest scale. So it's a change in allele frequency in a population over generations. Natural selection can change the distribution of a trait in one of 3 ways, and I think your book has some diagrams of these. So you have directional selection, causes a shift in a population's phenotype distribution. One of the extreme phenotypes that was once rare becomes more common. So it's moving in that direction. This type of selection is common when a population's environment changes or when members of a population migrate to a different habitat. So they move into a different habitat. So for instance, an example would be the increase in seed size available for food leads to an increase in beak depth in a population of the Galapagos finches. Then you have disruptive selection, which occurs when conditions favor individuals with both extremes of a phenotype range for individuals with intermediate phenotypes. So for instance, birds would either have small beaks or large beaks. They would not have the intermediate size. And then stabilizing selection will act against both extreme phenotypes and favors the intermediate variants. And so when stabilizing selection comes into place, it reduces variation. So for instance, most babies in the U.S. weigh between 6.5 and 8.8 pounds. Babies with much smaller or larger weights will have higher rates of mortality. Okay, there are some other factors that can affect evolution or have an effect on it. Gene flow is just the movement of alleles into and out of a population. It occurs because immigrants add alleles to the population, so those organisms coming in, and immigrants with an E take alleles away. So you can think of that E as exiting. Genetic drift is a random change in allele frequency in a small population following a chance event. So genetic drift shows up in small populations. So here's some effects of genetic drift. So it is significant only in small populations because it alters the allele frequency more than in large populations. So it would show up more in the small. It can cause the allele frequency to change at random. It can lead to a loss of genetic variation within a population, so it can do so by eliminating the allele from the population. And it can cause harmful alleles to become fixed, so that means they show up from then on. Now, there are 2 ways that genetic drift can show up. One is called bottleneck effect. So bottleneck effect is the genetic drift that occurs when the size of a population is reduced as by a natural disaster or human actions. So typically the surviving population is no longer genetically representative of the original population, so they have adapted to their new environment. The founder effect is where when a few individuals become isolated from the larger population, this small group may establish a new population whose gene pool differs from the source population or the original. You can also have sexual selection affect I lost my train of thought. Affect evolution. Sorry about that. And there are 2 types of sexual selection. You have intrasexual. Sexual selection is a form of selection in which individuals with certain inherited characteristics are more likely than other individuals to obtain mates, and it can result in what we call sexual dimorphism, which is a difference between the 2 sexes in secondary sexual characteristics, such as, now you need to fill this in, the size, color, ornamentation, and behavior of that organism. Sexual selection includes 2 types. You have intrasexual and intrasexual selection is selection within the same sex. Usually you'll see this more among males, where one male keeps the other males from mating with a female. So you see that a lot like in birds. Intersexual selection is also called mate choice, and the individuals of one sex, usually the females, are choosy in selecting a mate from the other sex. Usually they base it on the male's appearance and or behavior. Now, for this next section, Hardy-Weinberg equilibrium, you might want to have your calculator handy that has a square root function. Okay, So Hardy-Weinberg equilibrium describes populations that are not evolving. So this is typically based on a hypothetical population. However, it is common in natural populations for evolutionary changes to occur. But it's also common for natural populations to be in Hardy-Weinberg equilibrium for a specific gene at a certain locus. Plus, some populations evolve very slowly, so it's not easy to see these changes. It can be used as an initial test of whether evolution is occurring in a population. Also has medical applications, such as estimating the percentage of a population carrying the allele for an inherited disease. If the genotype frequency stays the same over time, the population is not evolving. It is said to be in Hardy-Weinberg equilibrium and must meet the following conditions. Number one, Hardy-Weinberg only occurs in an extremely large population. So the smaller the population, the more likely its allele frequencies will fluctuate by chance from one generation to the next. That process was once again called genetic drift. With Hardy-Weinberg, you're assuming there is no gene flow, so there can be no immigration, movement into or movement out of with the genes. In Hardy-Weinberg, there can be no mutations. Hardy-Weinberg, there has to be random mating, so there can be no sexual selection. In Hardy-Weinberg, you're assuming there's no natural selection, so all traits equally aid in survival. Real populations are rarely, once again, in Hardy-Weinberg equilibrium, but this model allows scientists to study how a population is changing. Okay, so we're going to talk about the Hardy-Weinberg equations. Hardy-Weinberg equations allow predictions of genotype frequencies and populations, but it's used only for simple dominant recessive inheritance. The values that the equations show are for a population equilibrium. Okay, so we have two equations, so let me rewrite them here. So we have p squared plus 2pq plus q squared is equal to 1, and then p plus q is equal to 1. This p squared is the frequency of the individuals with homozygous dominant trait. Q squared would be the frequency of individuals with the homozygous recessive, or who are, and then these 2pq are your heterozygotes. Okay? The p and q, when they're by themselves, p is your dominant allele. And q is your recessive allele. Okay. Okay. So it says use Hardy-Weinberg to calculate the predicted genotype frequencies for a population of fish, 1,000 fish where 640 have the dominant. Well, here's the deal. 640 with the dominant, some of those are going to be homozygous dominant, some are going to be the heterozygote because it's going to show the dominant. So what we want to do is we want to figure out how many have the recessive. So if 640 have the dominant, that means 360 are going to be q squared because that's your recessive, right? So we're going to take 360 over 1,000. So that's 0.36 is q squared. So if q squared is 0.36, then q, so you take the square of 0.36, not the square root, and so you get q to be 0.6. So if q is 0.6, then p is 1 minus 0.6, which is 0.4. So q is 0.6, p is 0.4. All right, so now we can plug these into this, okay? So our homozygous dominant allele will be 0.4, and we're going to square it. Is equal to 0.16, and then get it into percent. So times 100 is going to be 16%. Our heterozygote is going to be 2 times P, which is 0.4, times Q, which is 0.6. So we're going to take 2 times 0.4 times 0.6. So that's going to equal to 0.48, which times 100 will give you 48%. And then our homozygous recessive is going to be 0.6 squared, which is 0.36, which is 36%. Okay, so that is how you do find your percentages of your individuals that show a trait. So if I gave you a Hardy-Weinberg problem on the test, it'd probably be like for bonus. Okay. Okay, according to evolutionists, there are 5 factors that will lead to a population evolving, so genotype frequencies are different from those predicted. So some of these are going to be a repeat. So genetic drift. So genetic drift, once again, is the random change in allele frequency in a small population following a chance event. Gene flow. So if genes move into a population, then you can have a change in the population's genotype. Mutations. Although mutations work very slowly, so the effect occurs over a long period of time, so you won't necessarily see mutations real quickly. Sexual selection, non-random mating with own phenotype in individuals nearby, and then natural selection. Okay, speciation is just when you develop a new species, or a new species is developed. So it's a process where one species will split into 2 or more new species. Each time speciation occurs, the diversity of life increases. Now, there are going to be some things that keep species separate. So we're going to talk about reproductive barriers that keep species separate. So it keeps different species from interbreeding. So it keeps like a fish in a well. Let's say a fish and a sponge from interbreeding. It blocks gene flow and prevents formation of hybrids. So a hybrid is offspring that results from interspecific mating. It includes prezygotic factors. These occur before the zygote or fertilized egg occurs. So a zygote is a sperm that has fertilized an egg. So here are some examples. You have habitat isolation. So the species live in very different habitats within the same area and rarely meet. Then you could have temporal isolation. Breeding times are different among species. So breeding occurs at different times of the day or different times of the season. So some may breed in the spring, some may breed in the fall. Behavioral isolation. Different courtship rituals or other behaviors that prevent mate recognition between species. Mechanical isolation. The difference in physical structure prevents successful mating. So the male sexual organs would not accommodate the female, or the female might not accommodate the male. And then gametic isolation, the male and female gametes or different species fail to unite. You can also have post-zygotic barriers. These occur after fertilization and the zygote formation. So usually you're going to get a zygote to form, but it's not going to be viable. So you have reduced hybrid viability, so it's the development of or survival of the hybrid is impaired. Then you have reduced hybrid fertility. The hybrid fails to reproduce or to produce functional gametes. So for instance, in a mule. So a mule is a donkey and a horse, and so the gametes are not functional. And then hybrid breakdown. So the offspring of the hybrid are usually feeble or infertile, and so they don't make it to the age where they can reproduce. Okay, then there are several mechanisms that contribute to speciation. One is called allopatric and one is called sympatric. Allopatric speciation, your populations are separated by some geographical barrier, and so each adapts to a new environment. So for instance, a large lake gets divided into 2 smaller bodies of water, and over a period of time, the environments are different in those 2 bodies, and you get new species to develop. And then you have sympatric speciation. A new species will arise within the same geographic area as the parental species. And then you have coevolution. It's a process where 2 or more species evolve in response to changes in each other. So the species act as agents of natural selection on each other. All right, so that's it on evolution. So once again, I encourage you, if this is something you're interested in, you read your book more thoroughly. There's obviously plenty of articles online on evolution, different types of speciation, et cetera, that you are more than welcome to check out. Thank you.